At the outset of Blueprint, you identify a number of innate proclivities that would obviously bias our social interactions in certain ways. And a few that you mention in a cluster are in group favoritism, trade complementarity, social hierarchy, collective cooperation, network topology, social learning, and evolved morality. But before we
I
talk,
don't think I include trade complementarity and evolved morality in the social suite. So I'm not sure which list
it's
you're
not in the
alluding.
social suite. I'm not talking
Oh,
about that yet.
oh,
But
okay.
yeah, before we
Oh, I
talk
talk
about,
about the story about this, the story about when I was a boy playing with those Turkish kids. You mean?
yeah, that's what I'm
Uh-huh.
talking about.
Yeah, yeah.
But before we get into these items that I just mentioned, I'm just curious about the state of the investigation of the into the genetic basis of these traits in humans, because I recall from reading some of Trudy Mackey's work on the genetic basis of, I think it was mating dances and Drosophila, that these traits are so often polyvalent and highly complex. I mean, in that the genes that determine them are polyvalent, that it's difficult to pin down the actual genetic basis of the
Yes.
traits or the behaviors, even if you know that they're genetically determined. And this just makes me assume that since human behavior is, I mean, levels of magnitude, even though that's, I mean, a rough way of putting it, more complicated than the flies, the actual genetic basis of the behaviors that you're describing in the book, it must be very far from untangled.
Correct. And like any kind of, they're very, the most people when they think about how different variants of genes play a role in different phenotypes, remember the classic examples of thalassemia and sickle cell disease from high school biology, or the smooth and the rough peas, that Mendel looked at, which were, as you said, monogenetic traits, but in most animals, a given gene affects many traits. That's called pleiotropy, and when it comes to behaviors, there also not only does any given gene affect many different phenotypes. So, for example, I'm making this up. A gene that affects hair color might also affect anxiety. That's a completely made up example, but the same gene might play a role in both of those phenotypes. But also, any given phenotype has many genes that help explain it. So, as you suggested, it's a complicated mess. And in humans, we have very limited understanding. Now, we have some, for example, we have certain genes that are associated with schizophrenia. We have certain genes that are known to be associated with educational attainment, but explain only a tiny fraction of the variance. So most of educational attainment has nothing to do with our genes, but some of it has a little bit to do with our genes, and specific genes have been isolated that, or for which variants of those genes help explain such outcomes like schizophrenia and so on. However, having said that, there are animal models that are increasingly well worked out that are amazing. So one of the well worked out examples is some work with a certain kind of mouse species, Oldfield mice, that Hopi Hoekstra at Harvard has worked out. So these mice make a burrow under the ground and make a particular kind of burrow with an entrance and then a little a sort of cavern that they live in. And the kind of burrow they make is innate. So, like if you put the mouse in a cage and give it sand and wood chips and other natural materials, it'll just make this kind of burrow. Now, there's a different closely related species that makes a slightly different burrow that, in addition to certain other modifications to the neck entrance and the cavern part, has an escape hatch, builds a little escape hatch. And through some ingenious experiments where she and her students crossbred these mice and then isolated the genes that were responsible, was able to show that whether or not the mouse constructed a burrow with an escape hatch, this physical feature in the environment outside the mouse's body, depended, I think, on 2 or 3 specific genes. So if you had particular variants of these genes, then the mouse made a burrow with an escape hatch, otherwise it did not. So here's an example of an exophenotype under the control of a very limited set of particular genes. It's a complex behavior. It's shown in mice, And there are many other examples, not many, there are a handful of other such well worked out examples that I discuss in the book. And this, in combination with other evidence in humans, suggests that something similar is happening in humans. With other, we don't build burrows, we do other things. That's what's going on.